Okay, we are waiting a few minutes for people to join us. Good morning, Martin. I see he just joined. Martin is also a theoretical physicist in Paris, and he gave the talk last week. So he's back with us, or 2 weeks ago. Yeah. So just one more minute to allow people to join. Okay. Okay, it looks like the curve is flattening a little bit, but okay. So let us start then. Okay, there's still a few more coming. Okay, but let's start. Okay, so good morning, everyone. Welcome to this edition. Sorry, I don't know what I did with my screen now. Sorry, now I lost Zoom. Sorry. Okay, so welcome to this edition of Data at Breakfast. Today is very interesting because we have an intercontinental Data at Breakfast with a team that will share the talk. Yeah, and the team, the so-called QMATH team, is based at the University of Copenhagen and is traditionally, let's say, pre-COVID, a purely theoretical quantum computing, quantum information focused research group. And a few weeks ago, I posted on LinkedIn, here is my first paper in the Meta archive. Yeah? And a few minutes later, Matthias replied, and here's my first paper in the bio archive. Yeah? And so it was the perfect opportunity to invite them to share with us what a group of quantum computing physicists and mathematicians came up to fight the COVID pandemic. The team will share the presentation. I've been told that each of them will have, I guess, three slides. And they asked me to tell you to please, when you use the question and answer facility to ask the questions, maybe to indicate the name of the relative speaker, because he or she will then reply to your question. And Laura, who is a member of the team, will take the overall role of moderating a little bit your questions. So, Matthias and your team, thank you very much for joining us this morning, all the way from Copenhagen. And we are very much looking forward to your presentation. And if you would like to start sharing your slides, now would be perfect. Thank you.
Okay, thank you very much, Francesco. It is really a pleasure for us to have the opportunity to give this talk in South Africa. And for us, this has been a very unusual project, and we want to tell you a little bit about our project, both about how the project progressed and how and our results, of course. All right. So and as Francesco said, we are a purely quantum computing team. So in some sense, this was really a jump in the cold water for us. So my name is Matthias Christandl. And my co-authors are Andreas Bluhm, Fulvio Gesmundo, Frederik Ravn Klausen, Mančinska, Vincent Steffan, Daniel França, and Albert Werner. We will all speak for a few slides. And here you have our the information about our preprint, which we posted about 3 weeks ago, I think 2.5 weeks ago. So. All right, so let's get started. It all started in pre-corona times. We started a seminar series which we called Tensor Talk. We started in the fall, or what is the fall on here, half a year ago. every Thursday lunch seminar. And the topic of it was tensor networks. And tensor networks are actually related to, in other areas known as graphical models or hidden Markov models. And in quantum computing, tensor networks describe quantum matter, so the state of many particles of a quantum system. And there are two famous versions. One is called the matrix product state, and that is sort of like for describing one-dimensional quantum systems. So you see here a line of particles to describe the state and the behavior of that system. Or there's some other ones called projected entangled pair states that can describe these two-dimensional quantum systems. So we were interested in those, but we were also interested in their connections to other places in mathematics where they appear. And one topic is phylogenetic trees, so trees that describe the ancestry. And here you have an example of a tree that looks very similar to the others. So it's really just a graph on which this tensor network is expressed that is different. So that was a connection. And then Corona times came, and we moved our quantum information group meetings on Zoom. And maybe I should mention that in Denmark, the onset of the pandemic was in early March, and so we moved. The lockdown started on 13th of March. And we are a very international group, and you will today hear speakers from Brazil, Germany, Denmark, Latvia, and so on. We're very concerned about the corona situation, and we wanted to learn about the coronavirus, but we also wanted to use our abilities to make a small contribution to help. And so the project background was this unprecedented amount of public genomic data about the coronavirus. That is really unprecedented, and then the genome. We remember that the genome can be used to identify ancestry and phylogeny, and that of course connected to our tensor network experience, and so we set ourselves the goal. And here is a meeting from our first meeting here to build a phylogenetic tree of the Danish coronavirus. And you see here on Zoom where we discuss the relation of a human, chimpanzee, and orangutan, and the different phylogenetic trees that can describe the ancestry. So here the question is the chimp closer related to the human than to the orangutan, or in which way it is? So that was one of the goals, and another goal was to identify transmission chains relevant for Denmark for this. It was an unusual project for us because we come from the quantum area. We had to swap out our complex numbers that we usually use to real numbers. Formulas and proofs were changed to practical data analysis. Quantum information theory, we changed to bioinformatics, and of course you see a DNA string here on the right. And now usually our research is not time critical, or at least we think it's maybe not yet time critical because the quantum computer is still a few years off. But now we move to some area where it's extremely time critical, the research. It was really unusual for us. And the office we changed for live meetings on Zoom and the Zoom room. Other interaction and email went onto a collaboration platform, Slack, which was very helpful in coordinating us so we can work basically around the clock. Pen and paper, we changed to RStudio, which is this data analysis-focused programming language. And our normal work hours, I'm not going to talk about. And then we're really, when we work in quantum information, maybe there is a difference in experience from faculty to arranging to a PhD student, but here it was really different. We were all eight were complete newcomers to the subject, and we all had to learn it from scratch. So I want to give you an overview over the remaining talk. Laura will start to talk about the genome and mutations. Andreas will talk about phylogeny. Fulvio and Vincent will talk about data and data handling. Albert will talk about tree building. Frederik about big trees. And Daniel about transmission chains. And I will conclude. And with that, I hand over to Laura.
Yeah, so I'll tell you a little bit about the bio background and also set up kind of the terminology for the rest of the talk. So for coronavirus, just as for any organism, the instructions needed to copy and build it are written down in its genome. Now, this genome we can treat it as a long string. The next, Matthias, yeah. So we can treat it as a long string over the alphabet A, C, T, G, and we refer to these as nucleotides. In case of coronavirus, the string would be roughly 30,000 characters or letters long, and here you can see a short snippet of it. So the genome contains information needed to build proteins. So we have more than just 4 different proteins, so we need to use longer code words. And in the case of genome, 3 consecutive nucleotides, that is also referred to as codon, they encode for 1 amino acid. So proteins are built up of amino acids. So for instance, if we look at this short snippet here, we see that it starts with TAC, and that would code for this amino acid tyrosine, then it's followed by CCC, and that would correspond to proline. And this code, it is redundant, so meaning, any 3 nucleotides would code for some amino acid, but it is redundant. So there could be 2 different codons, these words of length 3, that code for the same amino acid. In particular, if you change the 3rd spot, then often the amino acid doesn't change, but that's not always the case. Here you can see an overview of the 30,000 long character string that corresponds to coronavirus genome, and we have marked different sections that correspond to different proteins. For instance, this 3rd protein is the spike protein. And in total, coronavirus has 10 different genes, so it corresponds to 10 different proteins. Now, as the virus multiplies in host organism, it gets copied over and over again, millions and millions of times. And of course, when we copy things so many times, mistakes can happen. And these mistakes are known as mutations. And one of the most common mistakes is substitution. So for instance, see the example on the right, there is a 6th letter, G, this nucleotide G, got mistakenly copied as T. And I've also written down what the corresponding amino acids are. And we see that in this case, the 2nd amino acid actually got changed because of this mistake in copying the nucleotide. And such a mutation would be known as non-synonymous because it results in a change in the amino acid. So, however, because this code is redundant, sometimes even though you copy a nucleotide wrong, the resulting amino acid doesn't change, and then such a thing would be known as synonymous mutation. And these substitutions, they are the most common type because usually other types of mistakes, such as deletions or insertions, they result in more drastic changes in the protein. Next, so here's a notation that we use to denote a specific mutation. So we record what position it occurs at, what was the original letters, and what did it get changed to. For instance, this example, we would write it down as G6T because the change happened in the 6th position, and originally it was a G, which was then substituted for T. Okay, but oh, and as a reference, we use this particular sequence from Wuhan, and this is very commonly done, that this particular sequence is used as a reference. Now, as this virus spreads, these mutations get passed on, and of course, more and more mutations happen. And one of the ways we could record the history of how these mutations layered on top of each other is using a tree. And Andreas now will tell you more about that.
Thanks, Laura. Yeah, so I will tell you about phylogenetic trees. And an example of a phylogenetic tree is the one to the right. As you can see, its leaves are not labeled with coronavirus sequences yet, but it's well, a collection of currently living primates, and to the very bottom, a mouse. So the mouse is obviously not a primate, so it should be very different from the rest. And the idea is that studying this tree, you can see the relationships between the different species. So, for example, and from the at the fourth leaf from up, there's the human, so there's us. And if you look at the tree, you see the next leaf to it in this tree is the chimp. So what does that mean? So it means that basically we suspect that there has been, like way back, there has been a different animal, which is the predecessor of both human and chimp. And if you go back, there probably has been an ancestor from which the human gorilla thing split from the human chimp split from the gorilla. And if you go back, you find the orangutan. So we are more closely related to the great apes than to the other primates displayed here. And then if you go way back, then maybe there has been like a species which we cannot see today, from which basically the primates split from the evolution of the mouse. So basically the mouse in our case will be the Wuhan sequence, because we know it's an early one, so it must be very different from the ones we see today. And we will substitute all the primates you see on the right, we will substitute by viral sequences. So instead of different species, we look at like different individuals from the same species. So if we go on, we can talk a bit more about the underlying models of this evolution. So now I just explained you the rough idea. And as I already said, now we'll look at trees where you have S1, S2, S3, so the leaves, the genetic sequences that Laura already introduced. So they will be of the form ATTA, GC, whatever. That's not a real sequence, I just made that one up. And so the first thing that we can think about is we can look at these sequences and say, okay, maybe the frequency with which A happens, with which T happens, G happens, and so on, will be more or less the probability of having this A, T, G, C at the leaves. So we will talk about probabilistic models, and in particular about Markov models. And there's two informations in the tree which are relevant here. One is the topology, so means like who is connected to who. So for example, S1 is connected to S2, but it's not connected directly to S3. And the second one is the edge length, so how long these edges are. But we are mostly focusing on the topology of this tree. The edge length, you can think a bit about time. So somehow it's time from basically the common evolution to the present. And we will assume, as you can see in this formula to the lower left, that this evolution is probabilistic. So there's a hidden transition or like a rate matrix Q that describes how much the nucleotides can change. So how, for example, has an entry how much A changes to T, and maybe it's different from the rate with which T changes to G. And the more time passes, the more of these changes can happen. That's why this TE, so the edge length is in there. Right. So on the next slide, we can see a concrete model. That's the easiest model that you can have. It's called the Jukes-Cantor model. That's not the one we're using, but it's good for illustration purposes. So there's two informations you need for this model. So one is the probability distribution at the root, and we assume that since we don't know any better, like in principle, the probability of having an A, the probability of having a G, and so on is always 1/4. So it's always the same. So we don't know what's more likely. So you can think about this as a Bayesian approach if you want. And now we need a model for the Q, for this rate matrix, and we will just assume that all the rates are the same. So all the entries, the off-diagonal entries will be like change from A to G, change from A to C, and so on. And we will always assume that this rate is alpha/3, where alpha is a parameter we want to determine. So if it's large, there's a lot of changes happening. If it's small, there's not so many changes happening. And this this model has the advantage that it's time reversible. So you can basically, that just means that you can measure distances between leaves. And if you do that, if you compute this distance, which is not so hard, you will find that the distance between sequences in this model is related to the Hamming distance between the sequences. So just you compare the sequences, and well, you always add 1 if you have a difference at one side. So that's the Hamming distance. I think Albert will talk about this later again. But and this is all the ingredients you need for this model. And in general, we will consider more complicated time reversible models, but to get the idea, the Jukes-Cantor model is completely enough. And yeah, then with this, I will pass to the next person, which will be Fulvio and Vincent, who talk about the actual data. So now basically the foundations part of the talk is over.
Yes, thank you very much. So next, we are going to start with the hands-on part and stop the theoretical part. And before we can do hands-on, we just need to clarify where we put our hands on, like what is our data. And in the ideal case, our data is exactly what Laura talked about. Our data is the sequence of the genome. And each of these sequences, as Laura said, should consist of 30,000 symbols. And these symbols are ACGT, but because measurements are, there are errors that can happen and ambiguities, we need ambiguity characters. So for example, the letter R would mean that the lab tells us that at this position there is either A or G. They are not quite sure. And this is for every combination. At the end we have an N. The N means that the lab knows that there's something at this position, but the lab doesn't know what is there. And on the right-hand side, you can see a short part, this really a very short part of one of these sequences for an actual coronavirus from Denmark. And where do we get this data from? This is fortunately very easy. There is a website which is a very good resource for these viruses. We download all our data from GIS8.org. And for our analysis, we use all data on GIS8.org, which is uploaded until May 26, which is then around 32,000 genomes from all over the world and 742 from Denmark. Each of these sequences has a label which tells us which lab sequenced this. This is the red part. And also on which collection date the sample was taken. And for some countries, there are additional metadata files which you can download, which give very valuable information like the travel history or the gender or the age of the patient, but sometimes also information like if the patient is still alive or if he died from the coronavirus. And this is not like this is not all you need. And Fulvio will tell you what we still have to do to this data you can download to be ready to build a tree with it.
Okay, thank you. And yes, so we get all this data from GIS8, and this data has mainly like two issues that can occur when the lab sequences the data or when they produce these strings. And so the first issue is that sometimes the lab is not sure, as Vincent said, it's not sure which character, which nucleotide is at some spot. And in that case, if they don't know, they put an N. So if a spot has an N, we don't have any information for that nucleotide. And the other issue is that sometimes the lab is not able to sequence the whole string for some patient. And there is some missing part, especially at the beginning and at the end. And these are marked with gap characters. So in those cases, like that part is not there at all. And this creates problems in sequencing, in analyzing these strings. And we only keep sequences of high quality. We want at least of these 30,000 character long, we want at least 29 of them to be there. and of 29,000 to be there, and of these 29,000, we want at most 300 of them to be N. In this way, we are sure that like most of the characters are there and are significant. And after doing this filtering, we are left of these 32,000 sequences that were there, about 20,000 are left, and of the 700 Danish ones, 582 are there. And now this is the set of sequences that we want to work with. And to do this, we still need one more preparation step, which is this aligning procedure. And so what happens when you have all this data? This is not aligned. Like if you see here, there is a snippet of a few sequences. Each line corresponds to a sequence. And you see that the first one, and then from the fifth one down, they are very well aligned. Like you see the same character in each column. But then there are like the second, the third, and the fourth, which are like all messed up. And why does this happen? Because in this case, especially at the beginning and at the end, there are missing parts of the genome, and like these sequences are just recorded in a file, and they are just all aligned at the beginning, and then one has to realign them to have corresponding characters stacked at the right position. And to do this, you reintroduce gap characters, and you realign everything. And in this way, in this new picture, you see that yes, the first and from the fifth down are still there, but then the second, the third, and the fourth have been shifted over by actually a lot, and you only see the very beginning of them, because say the second string was not really starting at the same spot as the first one, while it was starting all the way down when the 79 spot, and the same for something similar for the third and the fourth. And in this way, we have our sequences are aligned, and we can really compare them. And to do this like for us was very easy, because there is a package in R that is the cipher package that does everything for you. And now we have this aligned data, and this aligned data is ready to be analyzed, and is ready to build a tree. And now Albert will tell us how to do that.
Yeah, thanks a lot, Fulvio. And so let's just continue where we left off, namely with these aligned sequences. So that's the starting point, our input now, and where we want to go is the tree, which would look like this, and which would like resolve the different sequences, which are the leaves, for their common ancestry and how they are related. And the basic idea is what Laura and Andreas described, namely that sequences which are similar are more closely related than sequences which are less similar. So what we need is an idea of distance. So we have to introduce a distance measure, and what you can think about is, for the moment, just the Hamming distance. That is, we take two strings, we count the number of differences, and that's their distance from each other. And then we take, I mean, if we define a distance, we can compute the distance matrix between all the strings. So we take the pairwise distance, arrange this in a table, and based on this information, we want to build the tree. And the way we do this, I mean, you can think about this as a clustering procedure, where we try to find things which are like similar or more similar than the rest of the data that we have. Okay, and then how do we do this? So here's an example that we can do, one of these distance-based algorithms, UPGMA, which is an abbreviation for this complicated expression here. I'm not going to explain that one. So instead, let's look at an example for clustering or putting 5 different sequences into a tree. What you see here to the right in the table is the distance matrix, just a made-up example. So how could an algorithm work for this? Well, in the first step, we look for the sequences that are the closest. And I marked them here. So the closest sequences are A and D and B and C, because they only differ at two different positions. And then we say, okay, so that's a good indication that they should each of these pairs should have a common ancestor. So we put them in the same clade. That's what we call like this combination of A and D and B and C. Right. And on top of that, we like put the distance length in the tree depending on their individual distances. And then we just update the distance matrix. So we take this clade AD, for example, we compute the average distance from this clade to the rest of our sequences. And then we do this also for the second clade, the BC clade. Can you go on? Thanks. For the BC clade, and then we have this updated distance matrix. And then we just repeat. So we look again for the smallest distance. And in this case, it will tell us that E, the remaining sequence, is closest to the BC clade. So we also put this in and say, okay, E should have a common ancestor with the BC clade. And then we can complete now all the data is gone, and we can complete the tree and put like the final line in. And in that way, we end up with a tree. Of course, in practice, there are more elaborate schemes for this joining, which would not only take into account the closeness of the sequences, but also how far apart they are from, or how separated they are from the rest of the data, basically. So let's go on. So we get this tree, but how good is it? That's the next question one should ask. So here it's about quality assessment optimization. And the problem is, of course, that building such a tree is not necessarily unique. And so if you are used to different methods, you usually arrive at different trees. So the question is, which one of these trees is better? Which one should you use in order to infer something about the genome or the relations or the infection chains in the population? And basically, so there are different figures of merits that we could look at. We could try to minimize the number of mutations in the overall tree to explain our data. That would be a maximum parsimony approach. Or we could, taking into account the statistical models that Andreas described at the beginning, look for how likely it is to observe the given tree if we sample from such kind of data. That would be a maximum likelihood approach. Then we can take the tree that we built with one of these distance measures and optimize the starting tree with either one or a combination of these, and also see how robust a given tree is under different stability criteria. And then we do this, and finally we arrive at an end product, for example, using R, in our case, via the Fungon package. And here you see an example of Norwegian data. And in order to explain this picture a bit better and what we can infer from it, I hand over to Frederik.
Thanks. So now we've built our first tree, so let's try to take a look at it. We see in the picture, we have the tree on the left-hand side, and on the right-hand side, we have mapped out all mutations corresponding to this reference sequence from Wuhan that Laura also mentioned before. And you can see that the mutations, they determine the tree. For example, in the lower part, you have a rather long red mutation and blue, and they are the ones that separate the lower clade, whereas the upper clade is separated by these 4 blue, blue, green, red mutations. So there is this correspondence between the mutations and the tree, and when we have both, we have optimal information in some sense. Further, we have rooted the tree at the bottom with respect to the standard Wuhan sequence. So now we can start to, so this was an easy example of the Norwegian tree, but our goal was to build the Danish tree. So this we can do now because we can just run the algorithm with a different input. So that's on the next slide. And then we can start to make observations. So here we have the Danish tree, and there are significantly more Danish sequences than Norwegian available in this database. So it's maybe a bit more unclear what happens. So let me try to guide you through it. So what we some of the observations that we make is that around 70% of the Danish sequence, they have 6 very specific mutations compared to Wuhan. And further, together with those 6 mutations, there are more than 100 sequences that have an extra specific mutation, and they are the ones that determine the top clade. And this mutation, then you can check the entire database, where is it outside of Denmark, and that is primarily happened in Denmark. And Daniel will get back to this afterwards. And further, we also see that there are many different mutations in Denmark. There are these 70% that had these 6 specific mutations, but there were also many others that had many other combinations, which shows that the virus didn't just come from one place to Denmark, it came from many different places. And then once you want to try to determine which places it came from, you start to be more like of an investigating art. So let's try to investigate this a bit further. So now let's go back and look at the Norwegian tree again. Maybe you wondered why the Norwegian tree had names like Austria, Unknown, or Northern Italy. And this is not because these guys are from Austria, but these are Norwegians that went to respectively Austria and Northern Italy, and then we have the travel data on those sequences. So for example, the top sequence is a Norwegian person that went to Austria. And you see the 6 specific mutations that I talked about that were present in 70% of the Danish sequences. They are present in only the top 6, they are the 6, you see the blue, the green, the blue, the green, the red, and the pink sequences here. So they are not so common in Norway as in Denmark. And then maybe it's good to get some, just a little bit of background info. So Norway has snow and a lot of ski areas, but in Denmark we are close to the water, so we don't have snow. But so that means that many people in Denmark, they go skiing in a place where there's snow, for example, Austria. But the Norwegians, they have their own snow, so they don't go to Austria. So this is just some background info, and there was some talk also that a lot of people came back from Austria with infections and tested positive. And the question is, can we then see that in this data? So in fact, you saw that these 6 sequences, in Norway with the 6 mutations, they have the travel history of a transmission hotspot in Austria. So that somehow vaguely links the these 6 specific mutations to this transmission hotspot in Austrian ski areas. And if you then look, then you can start to look at more additional metadata. Then you can go to, let's look at the Icelandic sequences instead of just the Norwegian ones. And there you see the same match. And then you can try to do, now we made the Norwegian tree and the Danish tree. So if we try to do also the Austrian tree, you will see that this mutation is not that common in Austria, these 6 mutations, but in the ski area where you have the suspected transmission, the mutations are very common. So therefore, in this way, you can start to collect evidence that the 70%, that a large proportion of those, a large part of those, they came from one particular ski area in Austria. So, and now, as you can see, there's a lot more that you can view from these trees, and this Daniel will use this to track transmission chains now.
Thank you, Frederik. So, as Frederik was mentioning, we identify through the sequences and the trees and the metadata, we identify several transmission chains. So I'm going to exemplify how we do this with this one transmission chain. So these letters you see there, these abbreviations, they correspond to to different mutations, and this is one that happened within Denmark. So I'm going to show you how we inferred this transmission chain. So maybe we can go to the next slide. So if we go to this is again the big Danish tree that Frederik, no, I think it was actually, was it Frederik? Yes, Frederik showed to you a couple of minutes ago. So as you can tell, there is a lot of data in there. And this transmission chain we're going to discuss a bit more in detail now corresponds with this mutation on the upper left corner. So I don't know if you can see, but we're going to zoom in a couple of seconds. There is this one orange column over there. So this corresponds to this mutation C1302T. So these are all the Danish sequences which have this mutation, and we identified that it further mutated here in Denmark. So let's zoom into that column. So as I just mentioned, we start with this orange column over there, which with the green arrow. So this corresponds to C1302T, and as you can tell, it further mutated to this these sequences over there also have this further mutation C100074T, which is indicated by the blue arrow. And then if you go even further, there's this third mutation, which is there on the right, which corresponds to C2909. So and so on. So you can tell that we started with this one mutation on the left, and then this further mutated to the C11, and then to the one on the right, and this all happened within Denmark. So this is one example of how, for instance, you identify transmission chains that happened within Denmark. So maybe we can go to the next slide. And then we also saw the spread by looking at international sequences. We also identified the likely spread of some international transmission chains, which you can see on the plot on the left. So the virus also was very likely transmitted from Denmark to other countries. That's something we can conclude from our analysis. But what we also, maybe we can go to the next slide, please. We also identify some other strains that, for instance, this one, it most likely originated from the Netherlands, but it did, and then it spread to other European countries like Denmark. So doing this analysis, we identify several transmission chains within Denmark, but also internationally. Okay, and then I think this is all I wanted to say. Thank you.
Okay, so I will finish the talk by giving you some conclusions, summary and outlook. Well, for us, this certainly has been a very unusual project for a very unusual time. What we did on the science front, we identified transmission chains. So we showed our ability to do this using genomic data. We had to focus on some aspect. So we focused on Denmark because we're all based in Denmark. So we showed introductions of the virus to Denmark, transmissions inside Denmark, and the spread from Denmark in at least one case. Now, applications of this are, what we think are the most exciting applications is that these techniques can be used to assess mitigation strategies. For instance, they can be used for the identification of high-risk areas. Maybe that and as well for cross-check of contact tracing. So especially in an opening up phase, and one tries to via contact tracing to identify contacts and then put them in quarantine and so on in order to stop the spread of the virus. In order to let the rest of the country go on as normal as possible. Now, I will say more about, I want to say a little bit more about this, and this here is a graph from a study by from Iceland, Gudbrandsson et al. In the New England Journal of Medicine, which to a good part was a guidance and inspiration for our work. And what you see here is a graph from this paper, and you see a contact tracing network. So these are so if we focus on the middle where there's this person T48 that has the coronavirus, it was identified by traditional contact tracing that that person had been in contact with T68, T141, T107, etc. All right, with all the ones where you have arrows, it was identified by traditional contact tracing methods. This person has been in contact with, and you see the arrows and how it goes on. Now, the Iceland study has also sequenced the genome, and in this case, it's A2, A1. This is a plus 3 particular mutations, and so what they found is that that a lot of these places that were identified by traditional methods actually have this mutation, but when they did the population screening, they also found 2 others. So this person T48 likely also infected person T347 and T388, which you can now see as you can now see the ratio of them, for instance, as a measure of how good the contact tracing worked. Or in very specific other cases, you might imagine that you believe that you have caught everyone, but then you find a new sequence that you cannot attribute, and then you have to put an intensified effort in order to contact trace that person. And I think what is really interesting is that these genomic methods are able to identify these transmission chains and cross sectors. And we did our study on Iceland, but on Denmark. But one could also do this in a smaller setting, in a hospital, in other areas, etc., in order to help relief effort. Another application of this would be the study of the medical relevance of mutations. For instance, the study of the presence or absence of certain mutations, for instance, the spike protein, or a study of the overall abundance of virus strains. We've also looked into these aspects, but we cannot see any evidence for medical relevance of the mutations. Maybe we're also lacking some expertise on this area. But I think for us, the most immediate application would be to contact tracing and other non-medical interventions. With that, I would like to conclude the talk. I want to say thanks to a few people. Without this would have had possible. First of all, to Mads Albertsen's lab in Aalborg and Statens Serum Institute. They are producing and making available the sequences, and very clearly without this, our project would not have been possible. I also want to thank countless labs around the world for sequence data. Our paper has a 386-page acknowledgement table in a very, very small font size. I also want to thank a few friends we've bound this project off, to Anders Krogh, Carsten Bioff, and many others for feedback on the work. And as I mentioned already, I want to thank the Icelandic Genome Study, which provided the guidance. And our quantum group, our QMATH group, is sponsored by the Bluhm Foundation. I want to also thank them for their support. I want to thank you for listening, and Francesco for inviting us. And I have a last slide with the overview. So if you have questions, would be glad if you could address them to clearly to one member of the team. That will make it easier for us to efficiently answer. So thank you very much.
Yeah, thank you very much, Matthias, and thank you for putting up this slide so I can read off your names. It makes my life much easier. Thank you very much, Laura, Andreas, Fulvio, Vincent, Albert, Frederik, Daniel, and Matthias again. It was really an excellent talk and a perfect match for our Data at Breakfast series. I saw while you were talking that a few questions and answers popped up, but they disappeared. So maybe Laura,
yeah, I'll handle them. Yeah,
share with us the mystery of the questions. Yep, thank you.
Yes, so we have a few questions. So I think it makes sense to just take them as they came chronologically. So Marita was asking if our work has implications for vaccine development. And I told her that hopefully Matthias will touch a little bit on this. I'm not sure how much he actually did. So in some works, we can read that similar studies like ours can be used to inform vaccine design, meaning that by identifying places in genome that don't change much, so those can be used for vaccine development. But I don't know if anyone wants to say a few more words about that. I don't know, Matthias, do you want to say anything else?
No, I think maybe I was just going to express that we're open to discussing with anyone who's interested in this topic, but we ourselves have not, at least not yet, built up expertise in that area. So we do not know anything really about vaccine development, at least for my case, I speak for myself. But sure, it would be interesting to talk about it.
So Marita, do you want to follow up? Did we answer your question? Okay, I'm going to assume that the silence means that we've satisfactorily answered the question. So the next question is to Frederik, who was showing the trees that we built. And the question is whether our work implies or our analysis suggests that the virus started from Wuhan and then spread from there.
Yes, and we don't really investigate this. Somehow it is there as an input for the entire analysis. But with the code that we have written and put online, you can actually maybe do it slightly and investigate it yourself. But for us, this is not really a question that we ask and answer. Whether it's spread from, we can do many other places than Wuhan, but it comes from above that it started in Wuhan, because we take this reference sequence and then we map mutations with respect to the reference sequence. And somehow, if you pick the reference differently, then you might see something else. But it seems that everybody's picking this reference sequence, and it's very early, and it makes a lot of sense to it.
Yeah, Arun Kumar, do you have any follow-up, or was that a good enough answer? Again, I'm going to take silence as...
Yeah, Laura, sorry, the participants are all muted, so you can't expect to hear their voice.
Okay, they can't, okay, all right. So they just have to be satisfied.
You know, there are too many to allow everybody to speak, otherwise it would be...
Okay, fair enough.
Sorry for that.
We are in a very good position.
Yes, you have absolute control.
Yeah. So the next question is from Martin, and he's asking about viability of mutations. In particular, does the fact that certain mutations will not be viable and others will be advantageous have to be taken into account in the analysis? Or can natural selection be ignored in this kind of analysis? So I think we just observe mutations that actually didn't result in too drastic changes to the protein, because otherwise we wouldn't have gotten hold of those sequences. Yes, does anyone want to say something more about this question?
Yes, that somehow you could also turn it around and ask, can we see if there is some strain that takes over and which would indicate that it has an advantage? And in fact, we cannot see that at present, at least in the Danish data.
Yes, does anyone want to say anything more in this direction?
Yes, maybe. I would like to say a couple of words. So so we do. So you saw in the talk that we used Andreas explained this Jukes Cantor model, which somehow has an underlying assumption that all of the mutations from one nucleotide to another nucleotide, that they all have the same rate. But in our actual data analysis, we use a model where we allow different rates. So you can already kind of like, for instance, the T2C mutation. T2C is usually more frequent than certain other mutations. So that somehow takes into account a certain type of information about the viability of certain mutations, right? So the model in the maximum likelihood analysis do take that into account, and you could go to a more refined analysis. I think you could do such an analysis on an amino acid level or on other levels, but we use an arbitrary model for single nucleotide exchanges. I'm not sure if that addressed the question to some degree also.
Okay, so let's go on to the next question maybe, which is from Michael, who asks, can we continue to describe the viral sequence transformation as mutation or transformations? Mutations are supposed to be rare occurrences, but when it becomes a constant thing, can we continue to describe this as mutations? So I think this is just kind of some terminology that we use. Like, of course, at some point, this change happened in some sequence, and then for some reason it got passed on, and we can observe it in the population. It's not like it's constantly occurring again and again, as you say. I don't know if someone can help with this question. I'm also not sure I fully understood it.
Maybe we can also say that, I mean, the way we use this information about changes between or differences between the sequences is to infer ancestry and to identify infection chains, right? So it's, I mean, it's not our goal maybe to define like a new clade or like a particular subclass of the virus at that point, but more to extract the information here, how the mutations and the dynamics in a population happened at this point.
Mm-hmm. Yeah, Andreas, do you want to add anything?
I think everything has been said.
Okay.
No,
okay.
maybe I want to add something.
Yeah.
Namely the mutation rate, which I think is something interesting to say at this point. So, so people are now estimating that there are 10 to the -4 mutations per nucleotide per year. Okay, for these viruses, and maybe someone can give the correct estimate of how many nucleotides there are in this genome. I forgot the exact figure.
30,000, you mean?
30,000, yes. So by this you can do the back of the envelope computation of how many mutations you then expect for a given data set.
And maybe one should add that I think that the total number of mutations that we've seen is the total number of spots where there are mutations. This is around 300. Is that right?
Yeah, I think that's right. About right.
So there's only 300 positions out of these 30,000 where there are at present mutations, at least in the Danish data set. Is that also true for the worldwide, or how many do we have in the worldwide data set? How many spots in total? Does anyone know?
It's also around that figure, right, Fulvio?
Probably it's around that time.
Yeah, and a bit more. I guess it also depends exactly how many spots you want to say before it's a mutation, and how many times you need it to occur and stuff like this.
Yeah, so maybe let's take the very last question. So is it possible that this mutation can lead to a new strain of the virus? Who would like to take that?
Yeah, I guess for all of them, it's like every mutation, if it spreads enough, it becomes a new strain to be, yeah.
But I guess you could ask, maybe put more significance on this word strain, meaning that it has like different expression.
Yeah, and then I guess the answer is we don't know.
Yes. So I think our time is probably up, and we are also through the questions, unless I've missed something, or maybe someone wants to ask a new question.
Yeah, thank you very much, Laura, for moderating so nicely all the questions. I wanted to quickly ask Dr. Richard Lessels, who is part of the medical school of our university and has been involved with genomics of COVID all along. Would you mind, Richard, maybe sharing with Matthias and his team some of the activities in South Africa and Durban in this respect? Sorry to put you on the spot.
Sure, but I might do that by asking a couple of questions also.
Yes, yes, please, go ahead.
So I think this was a really great talk for some of us, again, where the phylogenetics is, we are involved with it, but we maybe don't understand all the background to it. This was really very helpful to go back to the basics. So one thing here is that, and one question I'll come to is around the sampling, and is around which samples actually get sequenced, and so which sequences you have to analyze, and what you can therefore infer from that. And one of the things here in South Africa, obviously, the ideal situation would be that you sequence every virus, every sample that tests positive in the country. And that would give you the most power to really understand your epidemic. But very few countries are able to do that. And actually, the United Kingdom are probably closest to doing that. And they just published some work. But also they're quite limited in understanding that genomic data because their epidemic was so overwhelming, and this huge number of introductions, similar to what you've kind of presented, that you had quite a large number of introductions by the looks of it, and then transmission. So the first question is just, do you have a good sense of which samples were being sequenced, and why were some samples sequenced and not others? So was it that clinicians or public health professionals were sequencing because there were outbreaks or clusters of infection so that they could understand that? Or was it somewhat more random that samples were sequenced? And then the second question is really about the clusters and the outbreaks. So one of the things that we are seeing very clearly here in South Africa is that our epidemic is still characterized by these clustered outbreaks in hospitals, in workplaces, and we may soon see in schools and universities potentially as they reopen. Is that something that you've also seen in Denmark, and is that something you've kind of been able to characterize with the genomic data?
Yeah, so maybe to start, we had some contact with the people at sequencing, but essentially I do not know the original reason for regarding your first question, right? For sequencing, so it seems to be mainly one lab sequencing in Denmark. That is Mads Albertsen's lab at Aalborg University. They are sequencing quite extensively, and you've seen that there were 700 sequences, but they are sequencing more, and we understand also that that there are more samples available also from the early times. But in Denmark, to give you maybe an overview over the testing and sequencing effort in Denmark, it was very varied. So in the there was testing and sequencing from the beginning, so that means early March. Though then from mid-March, so around the time of the lockdown, actually the testing, due to possibly capacity problems, testing was restricted. So testing actually not stopped, but like was only for mostly for patients in hospitals for about a month or so. So we suspect, and there's some informal evidence, that a lot of the samples are actually from very ill people, very ill patients, simply because the others were asked to stay at home and go in quarantine rather than trying to test everyone. And this strategy now over time has changed. And now the country has launched a more aggressive testing strategy in the, say, later phase of the Danish pandemic, and for the opening phase, where testing of population is encouraged, and also does not need like a medical referral anymore. You can just go and test, and I think then it's likely it will also be sequenced. This was to your first question. I don't know if someone wants to address more the second question.
Yeah, I just wanted to say that, so Richard, you mentioned these outbreaks you're having, like that we would actually be very interested to see whether we can apply our methods to kind of a more contained outbreak where we have where we can potentially have sequences from almost everyone, like in this outbreak, to see whether our methods, what would they say, and could we use them to kind of say something about how this outbreak originated. So yeah, if you, we would be interested to kind of keep talking to you if you had some ideas where we could obtain such sequences.
And what would be extremely interesting, I think, to just sort of like shoot for the sky, is if the genomic data was available during the outbreak in real time in a confined setting, because then we would really hope that we can in real time inform the relief effort. Okay? So say an outbreak in a hospital and you want to know, was it the receptionist or was it a patient? How did it happen? Because then you could therefore say, okay, now we have to change the setup in the hospital. We have to make sure this is changed so that this does not happen again, or so that we can avoid situations like that, or that we can stop a certain outbreak by this additional information. So I think this would, for us, be one of the most exciting situations in which we can see that our work has an actual impact, immediate in a relief effort. Because at the moment, what we showed you is sort of like our historic ability to identify transmission chains. And you might say, you somehow might say, so what, right? But sort of like the projection is, we showed this ability and we would like to use the ability. And in order to do that, and we do have somehow the data pipeline available, and we have the manpower in some sense, and we.
So essentially, that's what we are trying to do here. The challenge, of course, is that real time is tricky, because this virus moves exceptionally quickly, and one of the main hospital outbreaks that we investigated, we didn't really have the pipeline set up to do things in real time, and we came in quite late anyway to that outbreak. But we've kind of now got the pipeline set up to really generate the genomic data quite rapidly and analyze it very rapidly. And as I said, unfortunately, we don't have limitless funding in South Africa for this kind of research. So we have to try and focus the genomic effort on things like this, on high yield research that could really make a difference. And that's the intention, is to kind of be able to analyze these outbreaks to inform in real time the public health interventions.
Yeah, Richard and the QMATH team, thank you so much for this interesting discussion at the end, and I'm sure there is some interesting space for follow-ups, and we will do it probably in another setting. I'm a little bit concerned about the time because I see that our participants are leaving us because most of them have probably to face a day of work. So I would like to, at this point, to thank again very much Matthias and his team. Thank you very much to all of you for an excellent talk that opens up interesting questions and potential applications also in this part of the world. So we will definitely stay in touch. I will also thank Richard for briefly putting this in the context of of what is happening in our country here. So also a thank you to all the participants. We had almost 70, which is a very reasonable number. So thank you very much to all of you, in particular to those of you that follow us regularly. And some of you asked whether we will put it the talk online. We will do that in the course of the morning, of course. Yeah. So again, Matthias and your team, thank you so much for taking the time and sharing with us your interesting activity. And it's really amazing to see what quantum computing guys can come up with. Thank you very much. Thank you. We will stay in touch.
Thanks for having us.
Thank you very much. Have a good day and stay safe. Yeah.
Bye.
Thank you.
Thank you.
Bye. Bye. Thank you.
Bye. 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 Cheers.